This is a video to help you through the activity on osmosis and membrane transport. Before you begin this activity, it's a good idea to pause the video now and read through the information that's on the first page. Study the picture very carefully and read the information, and then unpause the video. One of the things we will talk about today is osmosis. And osmosis is movement of water across semi-permeable membranes, like cell membranes. Osmosis is a word that specifically refers to movement of water rather than other things. Later in this activity, we will talk about movement of other things across membranes. Right now, I'd like to focus on water. Here's a picture of a semi-permeable membrane in a beaker of water. The water molecules are shown by these little cartoons. If I separate two compartments of this beaker with a semi-permeable membrane, the water will be able to go through the membrane in either direction. There will constantly be water molecules moving from the left to the right and from the right to the left. When the rate of water moving in one direction is the same as the rate of water moving in the other direction, the levels of the water will not change. And there's no reason why one of these rates would be higher than the other. This is what is meant by dynamic equilibrium. It's dynamic in that the water molecules are always moving across the membrane, and it's in equilibrium because the overall amount of water on each side is not changing. The water solutions on either side of a semi-permeable membrane will be in dynamic equilibrium or will move to reach dynamic equilibrium. This is the case when there's only water on either side of the membrane. But what if there are solutions? Let's look carefully at these examples. In the beaker on the left side of this slide, I've put some solute molecules. The solute molecules are the green spheres. Notice that there are an equal number of solute molecules on either side of the membrane. And there is also the same amount of solution on either side of the membrane. When there are two solutions that have equal solute concentrations, these solutions have the same tonicity. Tonicity is a word that refers to the amount of solute in a solution. If they have the same amount of solute and the same tonicity, they are said to be isotonic. The prefix iso means same or equal, and tonicity or tonic means solute or the amount of solute in solution. So isotonic solutions are two solutions on either side of a membrane that have the same solute concentration. Let's look at a different example. In this beaker in the center, I have put solute molecules only on one side of the membrane. These are no longer isotonic solutions because one of the solutions has more solute molecules than the other side. This will change the equilibrium, and this will not be in equilibrium anymore, but it will try to reach equilibrium. There is more solute on the right-hand side of this beaker, and water will move towards the solute because it wants to dilute the solute. Another way to think of this is that there is a higher concentration of water on the left-hand side of this beaker and a lower concentration of water on the right-hand side. Water, just like other things, will try to move from high concentration to low concentration, or they move down their own concentration gradient. There are two ways to think of this. You can think of water moving from a higher concentration of water to a lower concentration of water, or you can remember that water will move toward the solute molecules, or it will move toward the higher concentration of solute molecules. The relationship between these two solutions is no longer isotonic. One side has more solute molecules than the other, or it has a higher tonicity. This is the side that is hypertonic. Hyper means more than, and again, the tonicity or tonic is referring to solute. A hypertonic solution is a solution separated by a semi-permeable membrane that has more solute molecules. The solution that has less solute molecules is hypotonic, and this is the solution with the lower concentration of solute. Since the water molecules are now going to move toward the solute at a faster rate than they will move back across the membrane, the water levels will change. So I'm representing that the water molecules are moving faster from the left to the right now than they are moving from right to left. Even though they're moving in both directions, it's a faster rate from the left to the right here, or toward the solute molecules. Eventually, you'll get this sort of a situation where you'll have more volume in the hypertonic solution because water has moved from the hypotonic solution to the hypertonic solution. This solution is still hypertonic and this solution is still hypotonic, but now there are different volumes. At some point, these solutions will reach equilibrium, and it will happen when the rate of water moving through the membrane in either direction becomes equal. The more volume you have over here, the more pressure there is on the solution, and the more it pushes water molecules back the other direction. When this solution reaches the point where the volume is pushing water molecules back through the membrane at the same rate as they're coming toward the solute, then this solution will eventually reach dynamic equilibrium. Water will travel through cell membranes just like they will travel through this semi-permeable membrane in a beaker. They can diffuse through the membrane by osmosis. Let's look at what happens to red blood cells when they're placed in different types of solutions. In the center is an isotonic solution. That means that the solute concentrations on the outside solution are the same as the solute concentrations on the other side of the membrane, or in other words, the solutions inside the cell. If this is the case, then water will move in and out of the cell through the membrane at the same rate, and you will have healthy red blood cells. Here's a cartoon of the blood cell in an isotonic solution, and here's an actual photograph of red blood cells that are in isotonic solution. If you put red blood cells in hypotonic solution, that means that the solution on the outside of the cell is hypotonic, or it has less solute than the inside of the cell. What that means is that water will move into the cell toward the higher concentration of solute. When water moves into the cell at a faster rate than it moves out of the cell will accumulate more volume and it will swell. Cells in hypotonic solutions will swell, and here's a photograph of red blood cells that are in a hypotonic solution. Depending on the actual tonicity of the solution, the red blood cells could either swell or they could break or burst. And this is called cell lysis when cells break down. On the other hand, if you put cells in a hypertonic solution, that means that there's more solute outside the cell than there is inside. The water will move from the lower concentration of solute to the higher concentration of solute. And in this case, it means from inside the cell to outside the cell. The cells will lose water, and they will shrink. This shrinking is called crenation. And here's a photograph of some red blood cells that have been placed in hypertonic solution. Let's look at a piece of vegetable and see what happens when you put vegetables in salt water or regular tap water. Here's salt water. This is a high concentration of salt, and it's a hypertonic solution. It's got a higher solute concentration on the outside of the celery than the inside. And water has moved out of the celery cells into the solution. This is what happens to celery when cells crenate. On the other hand, this celery stick is very crisp because water on the outside is hypotonic. Water will therefore move into the cells and make them swell, which makes the vegetable appear very crisp. Let's talk again about what this can do to red blood cells. On this slide, I've put 3 pictures of sodium chloride solutions. One is hypotonic, one is isotonic, and one is hypertonic in relationship to red blood cells. Red blood cells are isotonic with a 0.9% sodium chloride and or a 5% dextrose or glucose solution. So the center solution here is isotonic with red blood cells, and red blood cells would be just fine floating around in this kind of a solution. On the other hand, the 0.45% sodium chloride is hypotonic, and this is 5% sodium chloride, and that's hypertonic with relationship to a solution of sodium chloride that's isotonic with red blood cells. The 2 solutions on the outside would be dangerous for red blood cells. In some hospitals, you will be injecting any one of these things into your patients. But in some hospitals, the recommendation is to only use isotonic solutions. I'll tell you why. Here's an example of a solution that is injected into patients when they have low glucose levels in their blood. It's 50% dextrose. Red blood cells are isotonic with 5% dextrose. So this is a very hypertonic solution. Hypertonic solutions will cause crenation, and they can cause cells to die or lyse. Although it is standard procedure in many hospitals to inject 50% dextrose into patients that have low blood sugar, I'm going to show you what can happen to a patient when you inject a hypertonic solution into their blood vessels. This is an ulcer that was caused by 50% dextrose extravasation. Normally, when you inject a hypertonic solution into someone's arm, their blood will carry it away and dilute it immediately, and the 50% dextrose will not be that concentrated anymore. So if it kills any blood cells, it shouldn't kill very many. But if blood flow is poor, the dextrose or the hypertonic solution can pool in the bloodstream, and it can cause cell lysis. This is from the Annals of Emergency Medicine in 2006. And it was also reported in the Nurse ERR publication that is sent around to hospitals to warn of common mistakes. This article was warning doctors and nurses that hypertonic solutions can cause ulcers in their patients. And it was recommending that patients only be injected with isotonic solutions. The reason that this patient had poor blood flow was because he had a blood pressure cuff hidden underneath his hospital gown. So someone was taking his blood pressure and forgot to remove the cuff, and it was limiting blood flow to his arm. Unfortunately, this was the result. It's good to be aware of when you're dealing with hypertonic and hypotonic solutions and the potential repercussions so that you can take the appropriate precautions to prevent this sort of thing from happening. We just talked about osmosis, which is the movement of water across membranes. Now I'd like to talk about diffusion, which is the movement of solute from high concentration to low concentration. And solute in this case is anything but water. Here's a figure demonstrating diffusion. If you take food coloring and drop it into water, initially it is very concentrated and localized in one place. Here's a very high concentration of solute, the purple dye, and there's a very low concentration of that solute around in the surrounding solution. Diffusion is the movement of a solute from high concentration to lower concentration. This dye will diffuse to equalize concentration and make consistent concentration throughout the solution. Diffusion is movement of solute from high concentration to low concentration. Here's a picture of how diffusion can be applied to cell membranes. If you have a high concentration of solute, in this case the little purple balls, there's a high concentration of solute outside the membrane. It will move towards lower concentration on the other side of the membrane. If it can pass through the membrane, then it will simply do this. It will just diffuse through. Simple diffusion is the diffusion of molecules that can pass through a cell membrane. And there are only certain types of molecules that can do this. Small molecules can fit in between the cracks of the cell membrane, as long as they're not ionic or very polar. So most small molecules can fit in between the cracks. Molecules like oxygen, nitrogen, gases, carbon dioxide, and water. Even though water is polar, it can fit between the cracks, and it's not polar enough to prevent it from diffusing through a membrane. Other things that can diffuse through membranes are nonpolar things, because nonpolar things have no problem getting through the nonpolar barrier of the membrane. Remember what the membrane is. It's phospholipid bilayers, which have polar head groups on either side of the membrane, and it has a hydrophobic barrier or a nonpolar barrier that prevents polar things from getting through the membrane from one side to the other. So if things are nonpolar, they really don't have any trouble passing through this nonpolar barrier in between the bilayer. So things like lipids or other nonpolar molecules can usually get right through the membrane, even if they're pretty big. Things that aren't able to go right through the membrane are polar molecules, with the exception of water, and that includes ions. Ions are an extreme form of polarity. So ions cannot get through the membrane, even if they're very small. The other class of molecules are things that are really, really big. Things like proteins, which are much, much larger than the lipids or the triglycerides that we've seen before. Things that are very, very large have difficulty getting through the membrane, and so they need help to get through. They still can diffuse, perhaps, if they are offered a way to get from one side to the other. If they don't need help, this is called simple diffusion. But if they're still traveling from high concentration to low concentration, but they fall into the category here where they need some help, this is called facilitated diffusion. It's still diffusion because it's the movement of solute from high concentration to low concentration. And there are two ways that things can be facilitated through the membrane. One is through a protein that has a channel or a tunnel through the membrane. Here's an example. This cartoon here is showing a purple membrane protein that has a channel that allows solute molecules to travel through the membrane. Ions or polar molecules can get through the membrane through one of these channels. This doesn't require any energy, and it's still diffusion because it's the movement of solute molecules from high to low concentration. Another mechanism that solute molecules use to get through the membrane is through another type of protein called a transporter. This transporter can open on one side, gobble up a molecule, and then open on the other side and spit the molecule out. But this doesn't require energy if it's moving solute from high concentration to low concentration. Both of these types of proteins are membrane proteins that allow solute molecules a passage through the membrane. And in this case, when they're moving molecules from high concentration to low concentration, it is called facilitated diffusion. Once again, the things that need this are polar molecules, ions, and really large molecules. But what if you want to move molecules against a concentration gradient or from low concentration to high concentration? Then you need something else. And the most important aspect of this is that you need energy. Movement against a concentration gradient means to move solute molecules from low concentration to high concentration. This is the opposite of what happens in diffusion, and this requires energy. So when you're moving things from low concentration to high concentration, it's moving against a concentration gradient, and it requires energy. In the cell, energy is in the form of ATP. This is called active transport because it requires energy. Anytime you move solute molecules against a concentration gradient, it will require energy, and it will be active transport. On your activity, I've put this figure. This is a summary of the types of membrane transport that are related to solute molecules. The first three here are showing situations that are moving molecules from high concentration to a lower concentration. These are passive transports or diffusion because they don't require energy. If molecules can simply diffuse through the membrane without any help, they can fit right through the membrane cracks, or they can pass through this nonpolar barrier without any problem. This is called simple diffusion. If the molecules need help through a protein, they basically need a way to get through the membrane, but they're still being moved from high concentration to low concentration. This is called facilitated diffusion. The facilitation is offered by the protein. But once again, this does not require energy, and so it is passive transport, but it's facilitated diffusion. If you're moving solute molecules from low concentration to high concentration, like is shown in this figure, here I'm moving the square molecules from low concentration on the outside to high concentration on the inside. That requires energy in the form of ATP, and this is active transport. The important features of active transport are that you're moving things against a concentration gradient because that requires energy. Please try to answer the questions in your activity and check the key to make sure you're correct. Thanks for watching this video on osmosis and membrane transport. If you have any questions, please post them in the comments, and please vote if you like this video.